This is Dr. Randy Wolcott, and this is a presentation of biofilm infection. This is our first version, and when you clicked on this picture, we wanted to explain in detail at a level that just about anybody can understand what we envision is happening with a biofilm infection. First, we should point out that we do have a conflict of interest in that we do have a laboratory that does DNA diagnostic or microbial identification. But that laboratory also gives us the ability to see microorganisms very clearly on the surface of a wound or any infection. And we thought that just by knowing what microorganisms were present, we would understand what a biofilm infection is. But just look at the candlestick in this picture, and then I'd ask you to look at the faces in this picture. The exact same data, but if you organize it differently in your brain, it looks totally different, has different meaning. So the big elephant in the room for medicine right now is this biofilm infection thing, this second way that bacteria cause an infection. And whether it's a four-legged or a five-legged or a six-legged elephant, it is a big elephant. We'll try to show you why it's such a big thing. Well, the punchline is that medical biofilms, biofilm infections, are now well understood in the infectious disease community. This is a guidelines paper by 15 noted microbiologists and infectious disease specialists in Europe, and they conclude, as you can see down at the bottom, biofilms cause chronic infections. It's just totally different. You know what an acute infection is, you know what a chronic infection is, and it's microorganisms behaving in a way that we're pretty familiar with. We deal with biofilms every day of our lives, but they cause infection that we see clinically as chronic infections that are somewhat that are somewhat sensitive to antibiotics, but when you withdraw the antibiotics, they come right back, and that's what makes them chronic. So these specialists understand that biofilms are the cause for chronic infections. And if we look at all medical biofilms, the chronic infections where the authors have said this is a disease that's being caused by biofilms, such as chronic sinus infection in panel 1, or a chronic wound in panel 6, or infectious endocarditis, an infected heart valve in 8. Infected hardware of any kind in 13, cystic fibrosis, COPD, any stone like in panel 8, gallstones, kidney stones in panel 11, show that biofilm can infect any tissue of the body. And what's so disheartening is each one of these specialties have come to their own way of managing this chronic infection, but it's eerily similar. The ENT doctor, for sinusitis, or the cardiothoracic surgeon that replaces the valve will treat these chronic infections with episodic antibiotics, and when the tissue gets degraded enough that it no longer functions, the cardiothoracic surgeon will replace the heart valve, the ENT doctor will strip the sinuses, the lungs could be replaced, the hardware would be removed, and for chronic wounds we have the leg amputated. Now, we're not moralizing. That's the way chronic infections are dealt with in human disease in medicine right now. But once we understand biofilm infections, we think that that can fundamentally change. So let's talk about it. In a wound, microorganisms are present. That's universally agreed on. Now, those microorganisms that are present, whether they're bacteria, whether they're fungus, can do one of three things. They can have some positive effect on the wound. But what we know from a lot of detailed molecular science is that for something to be a commensal, there has to be crosstalk with the host. What that means is certain cells, like this dendritic cell, certain cytokines like IL-17, certain other molecules that are released by the bacteria that the host cells understand, all have to be in play. All those cells and those signaling molecules exist in the skin. Once you're through the skin and you're into the deeper tissues, subcutaneous tissue, muscle, tendon, it's all over. There are no commensal mechanisms at that level. What that means is all our normal skin germs like coag-negative staphylococcus or corynebacterium or propionibacterium acne, and there's a ton of them, when they're in a deep host environment, they are not commensals because those commensal mechanisms don't exist. There's neutral things that can happen, like this little contaminant here. This guy's on the surface of all this extracellular matrix. This is an acute wound, but this bacteria is not propagating. It's not causing any harm to the host, and in reality, there are contaminants, and we can see the contaminants when we go down and we look at a wound bed. But most wound beds look like this. There's groups of bacteria that are stuck onto the surface. You see the webbing material. Those are host proteins that are holding these bacteria onto the extracellular matrix. So that's host down in here, host extracellular matrix up here. This is fibrin, these fibers, but there's also rods. There's very small cocci. There's bigger cocci in other areas. So there's different morphology. So this is a polymicrobial infection that's stuck onto the surface of the host. And the question becomes, can that cause harm? Can that have a negative effect on the tissue that we call the wound bed? Well, the quick answer is biofilm infection has been around for at least a decade. What Costerton and Stewart were trying to show in Science, and that's a top-tier journal, is in a cystic fibrosis model, bacteria can find a host environment. Now, the white is planktonic cells, so individual motile bacteria find their way into a breach in the host epithelium. It attaches to the extracellular matrix. Now, if host defenses like white blood cells, antibodies, or even if there's antibiotics on board, if they don't kill that bacteria and it's able to reside for even 20, 30 minutes, we see panel B happening, where there's a transformations like a caterpillar to a butterfly, where the bacteria expresses a third of its genome, a third, like 800 new genes are expressed. So it makes all these different proteins, all these different materials. It self-secretes a matrix, and this early structure called a microcolony is now walled off from antibodies, walled off from white blood cells, so host immunity can't get to it anymore. And it doesn't take very long for this to happen. Once it's protected in this early matrix material, it starts growing. Now, these cells are metabolically active. They'll grow and they'll fill the space. If there's not enough space, they'll incite inflammation of white blood cells to cause collateral damage, and then that'll gather more space for the chronic infection to exist. So it's pretty nice. So you've got bacteria that attaches, it forms the matrix material, it grows, and we'll talk about some of the structures inside, and then it feeds itself through inflammation. If it was to digest and kill the tissue that it was on, it would be lost to the environment. So it has to inflame the host and eat the exudate that comes out from the capillaries. And that would be a biofilm infection. At a molecular level, this was a review in a cell-hosted microbe by Kim, and he was dealing with some of the controversies of the day. And the controversies were that a third of experiments were showing that certain bacteria, when they came in contact with epithelial cells, would breach those cells, digest those cells, and then would propagate that way. And he called that a breaching behavior. And many experiments showed that, dozens and dozens. If you take those same bacteria, the Shigellas, H. pylori, Salmonella, E. coli, and you put them in different conditions with the same epithelial cells, they'll attach to those cells. They will take over the cytoskeleton of the cell. They'll block apoptotic pathways. They'll upregulate cell survival pathways, and they'll prevent the cell from dying. So it immortalizes the cell, and he called that colonizing behavior. We would call that biofilm behavior. Well, I like this panel. Panel A looks like an acute infection. That's everything we learned in microbiology that defines an acute infection. B looks like a biofilm infection. Bacteria that are sessile, that are attached, that commandeer host function so it has a secure substrate to be attached to and maintain its biofilm structure. Now, Kim called this a new paradigm in bacterial pathogenesis, just a fancy way to say this is a second way that bacteria infect. And that is indeed the case. That's what we just showed in the Costerton-Stewart slide. So we come up to our drawing. This is from Rick Postal, and I gotta give him a shout out. This, I think, is an excellent drawing. And so what we'll do is we'll just break it down. We'll talk about the different aspects of this drawing and what we're trying to communicate. Many drawings, like you saw with the Costerton-Stewart or the Kim, show the bacterial side of this. What we wanted to do is bring in the host functions, the host relationships that truly define a biofilm infection, and why what we see clinically in a chronic wound is really a chronic infection. First, we have to prove that a chronic wound is a chronic infection, and we turn to Koch's postulates for this. Koch's postulates fell out of favor for a few years, but in simple microorganisms that don't require any vectors or any strange things to happen before they can infect, transferring that into an animal model that can demonstrate the infection is just simplistic, and it's easy, and it's a good model to use. What we found in our mice is, now we've done over 40 mice, is that all but one will show that when we seed the slough or the microbiota from a chronic human wound into this mouse model, we get a glistening slough that forms exudate, and the most important thing is the wound healing is stalled. In a non-infected, the wounds that we don't seed with the chronic wound microbiota, those wounds heal up in 8 to 12 days. There's no slough, there's no exudate. So we can reproduce the chronic wound in the mouse. We can show the harm from the microbiota. That does demonstrate conclusively that the wound microbiota causes the chronicity, causes the chronic wound. So the cause and effect is there. In evidence-based medicine, it requires more. This is Hill's criteria for causation, and you can see that the coherence or the laboratory would be Koch's model, but all the rest of these things can be easily explained from the literature. Association just says, are there microorganisms present? So the causative agent has to be associated with the lesion, and it is 100% of the time. Specificity, is there a specific reason why we see what we see? If you use host explanations, well, there's poor circulation or there's pressure, etc., then you can't explain why, if we biopsy through a wound, or if we biopsy by a wound, or if we get a tape tear or a traumatic wound next to a chronic wound, those things all heal up in 2 weeks, whereas the chronic wound keeps going. So it can't be host deficiencies. The best local explanation is the microorganisms. Reproducibility, if you're going to create animal models for a chronic wound, almost every animal model to produce that, whether it's in a rabbit ear, whether it's in a pig model, whether it's in a mouse model, these animal models require seeding with microorganisms to produce those chronic wounds that they test the different products against. Temporality just means that there has to be an association in time. So shortly after you do your microbial seeding, or you give it the microbial challenge, there has to be some changes that can be related back to the microorganisms. And in the seeding of our animal models, that is indeed the case. Biologic gradient, we all know this. If you have a highly contaminated wound, you tend to do more antimicrobial strategies because bacteria, the microorganisms, tend to cause more harm. So that's the gradient. The more there is, the more effect you see from that causative agent, which would be the microorganisms. Plausibility, does it make sense that the microorganisms could stall healing and cause what we see with a chronic wound? And we'll show you more about that in the next slide. But yeah, the biochemistry of chronic infections is well worked out, and it's the same. There's elevated pro-inflammatory cytokines, there's elevated proteases, there's elevated neutrophils, and a host of other things that we see in all chronic infections, and chronic wounds have those things. Experimental data, animal models, laboratory in vitro models, cell culture models, all suggest that bacteria, sort of like we saw in the Kim article, that's a cell culture model, that the bacteria can produce these colonizing effects, what Kim called it, or biofilm effects, which we would call chronic infections. And then analogy, do we see any other host disease that looks similar? Well, chronic infections look like chronic wounds. They wax and wane. They're resistant to antibiotics. You withdraw the antibiotics and they come right back. They're highly inflammatory. They got the same biochemistry. Yeah, the analogies there. Chronic wounds behave and look like chronic infections. So you don't need all of these Hill's criteria to say that there's a cause and effect, but the fact that all of them are pretty obvious says that the microbiota in biofilm phenotype is the cause for chronic wounds. I promise this slide, basically what this is showing is that biofilm structures and functions can easily explain what we're seeing in the chronic wound. The biggest thing is it's stalled. Well, why is there stalled healing? Well, there's multiple reasons. Hyperinflammatory mechanisms will cause damage to host healing mechanisms, or it will just commandeer cells that are needed for the host healing process and keep them on the inflammatory side. There's diffusion of small molecules like we showed before that take over the cytoskeleton, that take over apoptotic pathways, that take over multiple other things that do not let the host cells do their normal activity to participate in healing. Fibroblasts can't become myofibroblasts and cause wound contraction. Keratinocytes can't migrate. So I mean, the list goes on. So the structure and function of biofilm can clearly be the best reason for all the chronic wound behavior that we see clinically. So we're back to biofilm infection. We'll try to explain this. It's a busy slide. So what I'd have you look at first is the blue portion, the matrix portion, that shows the dragon, the octopus, the alligator. So what I asked Rick to do was just, there's mechanisms by which the matrix does not allow harmful things to get to the individual cells working within the substance of the biofilm. So it can delay diffusion. It can react to reactive substances that are trying to get in, like peroxide or other oxidizing or reactive molecules. And then there's mechanisms we don't know. The other thing that the biofilm matrix does is it provides a boundary function so that the communication molecules, the quorum sensing molecules, can reach and congregate and affect the different species of bacteria that are co-localizing within the substance of the biofilm. Now, anoxic cores we won't deal with much, but these regions where all the oxygen gets used up by the busy bacteria will cause a lot of bacteria to go dormant in that area, but it also provides an environment for strict anaerobes, bacteria that we would never expect to see in a wound environment, growing and being a huge part of the relative abundance of bacteria in wound biofilm, up to 25%. So these strict anaerobes add a lot of important things to the infection. They can allow other bacteria to be more pathogenic, and they can be pathogenic in their own right. So the anoxic cores are very important, but it's how this entire system functions to produce human disease that we really. What we want to look at first is the matrix, that moat, that blue moat that we had drawn around the biofilm. And what white blood cells should do when they encounter bacteria is phagocytize it. And this, you saw that said slippery capsule coating was around the bacteria. That is the biofilm matrix. That's the extracellular polymeric substance that bacteria puts around itself. Now, there's only two bacterial cells in this group, but it's clear that the white blood cell can't get to it. And here is bacteria that's stimulated by a PAMP, or a pathogen-associated molecular pattern, and they'll go to that PAMP. But just envision that being this biofilm on your right, and they get down, they get up next to the matrix material, and then they just sit. They're trying to get to the bacteria, but the matrix material precludes them from reaching that bacteria and phagocytizing it. So let's talk about what this biofilm does as a whole. Those individual bacteria cells that are working synergistically inside diffuse or inject small molecules out into the host environment, and the cells take up these small molecules, effector proteins and others. And the first thing we see is that you saw in the Kim diagram was pedestals, the cytoskeleton reorganizing. Regardless, when the biofilm takes over the internal functions of the host cell, that host cell can't migrate. That means it can't put out little pseudopods and work its way over. It can't shed. It can't release gap junctions, which are part of the cytoskeleton, and release out into the environment, and therefore it can't get rid of the biofilm. And it can't divide. The process of mitosis requires a functional cytoskeleton. So the effector proteins have attacked the host cell at one of its most fundamental levels, and won't allow that cell to function the way it's supposed to, like a myofibroblast can't contract the wound. So this is a real important step. The biofilm has to be functional at this step, or it can't cause a successful chronic wound. Senescence, you can see the pedestal formation. This is a type 3 secretory system. This E. coli injects molecules into the host cell, an epithelial cell. This could be a wound bed cell, and forms this pedestal, and this is biofilm. It's attached bacteria. You don't see any matrix around it yet, but it will come. But attached bacteria is biofilm. And again, this is one of the most important processes that the bacteria does. The other thing that the bacteria has to do is it has to prevent the cell from dying, and you'll see that in a little movie here in just a second. It creates zombie cells. These are cells that are alive. They have metabolic processes occurring inside the cell itself. But it can't apoptose, it can never die, and it can't communicate with the other host cells in the area normally. And the cytoskeleton's gone, and manufacturing is curtailed, and there's just a lot going on. But if the biofilm is not successful in making this a zombie cell, then it's released into the environment, and it's not an effective chronic infection. So the main pathways that are reported that this type 3 secretory system attacks is this caspase system. It attacks it at multiple different levels, and then there's other apoptotic pathways that are blocked by these different effector proteins. They go after ubiquitin, they go after things that we don't even know about yet. This list is now far larger than what you see on the right, and you see a lot of unknowns in those different categories. But a lot more effector proteins and other secretory molecules have been identified. Here's a review that's not even 2 years old, just showing you that it's growing. We understand more and more about these subcellular pathways, E. coli, Salmonella, Shigella, lots of other bacteria. But again, what they're showing is they impair these and prevent these apoptotic pathways from functioning at all, and that's important. Now, why? If Shigella or Salmonella or E. coli can inject these molecules into the cell, prevent this cell from dying, from popping, then they have a stable base from which to form the biofilm, what Kim called colonization, what we would call biofilm. So I'm going to show you a movie, and if you have a wild type bacteria that can put some of these molecules in and prevent apoptosis, that's successful. Then it'll show you a knockout where the OSPC3 is not present. It's just one of these effector proteins. It does not block the apoptotic pathway, and the host cell is able to die. So watch those. This is Yersinia, these little black dots. They're attaching to the host. They're injecting the OSPC3 into the cell, and they're successful. This is the knockout model now. And you see that the cell apoptoses and dies, and you see that the bacteria stops, and it's going to be released with the dead host cell. So that one effector protein is the determination whether that infection, that biofilm, gets started in that host or not. So these are targets. These are things that we can look for dealing with biofilm infection. Now the big thing is inflammation. All chronic infections are associated with persistent and hyperinflammation, not 10 times, but 100 times, 1,000 times what you would see in a normal lesion. So if you have a normal wound, like a surgical wound where bacteria is not involved, it goes up just to a very small amount. You get bacteria involved, and then you see, again, 100 or 1,000 times more neutrophils, more pro-inflammatory cytokines, more matrix metalloproteases. So why is inflammation so important? Well, how would the biofilm feed itself if it can't kill the tissue and digest the tissue that's under it? Well, it has to inflame the capillary bed that's underneath the wound or at where the chronic infection is. Plasma leaks out, and that plasma is a constant, sustainable nutrient source for the biofilm. Now the biofilm takes on a parasitic characteristic. It's a parasite. It doesn't kill the host. It self-regulates its size. It cross-communicates with the host. And through that inflammation, it will exacerbate the host when it needs more plasma. And then when it wants to go quiescent, it goes quiescent. That's the waxing and waning you see in chronic infections. It's just the biofilm deciding it needs more nutrients or less nutrients. So inflammation is a critical part of the life cycle and the success of how a biofilm causes infection. It's important to note that if we add steroids to lesions like a chronic otitis externa or a chronic infection of the eye, when you add the steroid with the antibiotic, it's more effective, because you're diminishing the nutrient source to the biofilm. Now, adding steroids to a chronic wound, you also neutralize host healing mechanisms. So that's like a sum-zero game. Yeah, you diminish the biofilm, but now you're also taking away host healing properties. So the steroids in chronic wounds may not be that effective, but just to point out that inflammation is a key component to the life cycle of a biofilm infection. This shows a lot of different things. The photomicrograph shows what Dr. Diegelman was trying to show is lots of neutrophils in the blood vessels, in the tissue, and there's thousands of times more neutrophils in this wound bed histology, biopsy from a wound bed. But then also just right above this tissue, you see all this cell density, and that is a clear biofilm that is kind of investing itself down into the tissue. But this biofilm is releasing substances that call these neutrophils in, and then as Dr. Hartle shows in his research, this is from cystic fibrosis, but it's similar. The neutrophils are brought in by different mechanisms. Once the neutrophils are there, soluble type 4 secretory molecules impair those neutrophils, commandeer the internal function of those neutrophils, and they can't migrate anymore. So, but they also can't apoptose, and they just sit around until their membranes break down and they degranulate. When they degranulate, they release elastase and MMPs. That causes a trapping of more neutrophils, the release of these fragments. Those fragments cause hyperinflammation, brings in more new neutrophils, and then the cycle repeats itself. And that cycle's outside of host control. The host can't downregulate that, so that's how you get to 100 times, 1,000 times the amount of pro-inflammatory cytokines and the levels of inflammation that are detrimental to the host. So this is well-researched, well-demonstrated property of biofilm is to produce this inflammation for a source and to prevent the host from healing the wound. Now, a lot of the things that we've said in this biofilm infection lecture may not be true in 2 or 3 years. Right now, I really believe that what we're saying is true. We do know, however, that from what we've seen, from the way the wounds reacted to our assumptions for treatment, that biofilm is an important barrier of chronic wounds, and it probably is the cause of chronic wounds. So in general, I don't think anything's going to change. Now, these specific things like inflammation and cytoskeleton and stuff like that, yeah, we're going to grow. We're going to learn more as we go along. But we've started down the slippery slope. What you need to take away from this is that bacteria have 2 ways they can infect a host. The acute infection, which you've always known, that's what you've been taught in microbiology, but chronic infections are caused by biofilm phenotype microorganisms. Thank you.